Assalamu alaikum and welcome to the medical concept with Dr. Falak. Today we will be discussing about another very basic physiological concept, the blood and its composition. Have you ever thought why is blood red, but the fluid part of the blood is actually yellow? And what happens if just one component of the blood stops working? Why can that become life-threatening within minutes? So today we are going to break down blood not just as a fluid but as a highly specialized connective tissue that is essential for survival. In this lecture we will going to cover two core aspects. Number one is the composition of the blood, means what exactly the blood is made up of, and second, the functions of the blood and their component, means how each component contributes to maintain the overall homeostasis of the body. So by the end of this section, you should be able to connect structure with the functions, which is exactly what you will need in your exams and even for clinical understanding later on. Now look at this picture. This picture is known as peripheral blood smear. And what is this peripheral blood smear? It's a thin film of the blood that is spread on a slide, stained commonly with right stain or the Giemsa stain, and later on examined under a microscope to evaluate the morphology and the distribution of various blood cells. In this picture, you can easily appreciate these red circle biconcave structures. And what are those? These are actually the red blood cells. You will also find another type of cells that are slightly bigger than these red blood cells. This one, this one, this one. They are different type of cells and they are referred to as white blood cells. You will also going to appreciate these small amount of dots that are bluish dot here, that appear bluish because of some type of stains, and these small fragments are known as the platelets. So that's how a peripheral blood smear looks upon the microscope. Now coming to the blood. What is blood? Blood is defined as a specialized type of connective tissue that is derived from the mesoderm. It's mostly composed of two main components, the plasma, that is approximately 55%, and the formed cellular components or the formed elements that forms approximately 45% of the total blood. They include red blood cells, white blood cells, and the platelets. Now coming to the properties of blood. Blood appears as bright red in the arteries because of the presence of oxygenated hemoglobin, while dark red in the venous blood because of the presence of deoxygenated hemoglobin. The total volume of the blood in adult male is approximately 5 to 6 liters and in females 4 to 5 liters. That makes 7 to 8% of the total body weight. Blood is slightly alkaline in pH with normal range 7.35 to 7.45, while the temperature reflects core body temperature, that is 98.6 degree Fahrenheit. Blood is 3 to 4 times more viscous than water, while specific gravity of the whole blood is 1.05 to 1.06. Osmolarity is 285 to 295 milliosmol per liter. Regarding exam point of view, you have to remember all those numbers that include the osmolarity, the specific gravity, the viscosity, and its pH. These are all very important, okay? And also the volume of the blood in males as well as in females. Now coming to the composition of the blood, as we have already discussed, that blood is composed of the plasma and the formed elements. Now if we look into the plasma, plasma is further divided into various components. It's mostly composed of water, that is 90 to 92%, the proteins, that is 6 to 8%, and the various solute that makes approximately 1 to 2% of the total plasma. Then comes to the formed elements. Formed elements include red blood cells, white blood cells, platelet, and certain cell fragments, while white blood cells can be further divided into the granulocytes and the agranulocytes. This whole classification is very important because it will give you a big picture and idea about the composition of the blood. Now tell me the difference between these two test tubes that are containing blood. Are you able to appreciate? This one is the plasma and this one is the serum. So what is the difference between the plasma and serum? And this is a very important exam and the viva question. Now let's see. What is the main difference between the plasma and the serum? When we talk about the definition, the plasma is the liquid part of the blood with clotting factors, while serum is the fluid that is obtained after the blood has been clotted. Means it is devoid of clotting factors. This is the main definition that will guide you. Regarding clotting factors are present in the plasma, including the fibrinogen, while they are absent in the serum. Regarding method of preparation means how you are going to obtain the plasma in the serum. First, blood is added up with the anticoagulant and then centrifugation is done so that they will lead to the plasma. While for the serum to obtain, first blood is allowed to clot, later on centrifugation is done and then we are going to obtain a clear serum. Fibrinogen is present in the plasma while it's absent in the serum. Appearance. The plasma appears slightly turbid because of the presence of clotting proteins, while serum appears very clear because of the absence of clotting factors. Volume. Plasma is slightly higher in volume as compared to the serum. And what are the uses? Plasma is used in the coagulation studies, example prothrombin time, activated partial thromboplastin time, and in the case of transfusion, while serum is mostly used in the biochemistry test, serological test, and hormone assays. Anticoagulant needed? Yes, anticoagulants are needed in the plasma and then there is no need of anticoagulant in the serum. So this slide is very important. It will give you a clear differentiation points between the plasma and the serum. Now coming to the main components of the blood. First, we are going to discuss the red blood cells that are also known as erythrocytes. Erythro word is for red and cytes is for cell. Regarding definition, the erythrocytes are anucleated biconcave blood cells. Anucleated means that they do not contain nuclei. These cells are specialized for the transport of respiratory gases that include oxygen and carbon dioxide with the help of hemoglobin. This red blood cells, they are the most abundant formed elements in the blood. Approximately, we have just discussed the total count is 5 to 6 millions per microliter in the males, while in females they form approximately 4 to 5 million per microliter in females. The total lifespan is about 120 days and very important MCQ. And these red blood cells, they are destroyed mainly in the spleen. Now tell me, why males are having more red blood cells as compared to the females? Can you answer this question? This is because of the testosterone-induced erythropoiesis in the male, higher metabolic demands in the male, and the absence of menstrual blood loss in the male. So that's why you will find more red blood cells in the males. Now coming to white blood cells. White blood cells are also known as leukocytes. Leuko word is for white and cyte word is for cells. These cells are nucleated with irregular or amoeboid shape. They are mostly involved in the body's defense system and immune response against infection and the foreign substance. They are less numerous than red blood cells. And in size, they are larger than the red blood cells and their range is approximately 10 to 20 micrometer. The total count is approximately 4 to 11,000 per microliter and the total lifespan varies depending upon their action. It could be hours to days for neutrophils and it could be months to years for lymphocytes. There are two types of white blood cells, granulocytes that contain granules in their cytoplasm and agranulocytes means those white blood cells that do not have any type of granules in their cytoplasm. So again for this white blood cells you have to remember their total count. This one 4,000 to 11,000. This is very important and even the lifespan is important. And what are the different type of granulocytes and agranulocytes? So let's discuss. Regarding granulocytes you can see that we have neutrophils, eosinophils and basophils. So what is neutrophil? This is the main typical appearance of the neutrophils. They are multi-lobed nucleated cells and have pale lilac fine type of granules in their cytoplasm. Features: they stain poorly with acidic and basic dye, so that's why they are known as neutrophils. Okay, means neutral cells. Phil word is for loving and the first word was for the stain that they are going to pick. What is the main function? They are responsible for the destruction of the bacteria by the process of phagocytosis. Then comes eosinophil. Look at this image of the eosinophil, right? These cells are bilobed in their structure, means their nucleus is bilobed, and they contain granules that are bright red to orange in color. They mostly stain with acidic dye and they are important for provision of defense against parasite, and they modulate allergic reaction as well. Now third type is the basophil. This is the typical appearance of the basophil. Basophils are actually having bilobed or S-shaped nuclei, as in the picture is showing. They are having large deep blue to purple granules in their cytoplasm, and because of these large granules the nucleus is not clearly visible. They stain with basic dye, example methylene blue, so that's why they are known as basophils. Regarding their function, they are responsible for the mediation of allergic, especially type 1 type of hypersensitivity reactions by releasing of the histamine and heparin. Now coming to the agranulocytes. They are of two types, lymphocyte and the monocytes. Lymphocytes are having a large round nuclei with a thin rim of cytoplasm, and they are considered the smallest white blood cells. They are of two types of lymphocyte, B cells and the T cells, and the third type is the natural killer cells. They are having a specific role in the adaptive immunity. Beta cells are responsible for the production of the antibody, while T cells are important for cell-mediated immunity, while natural killer cells are important for cytotoxic reaction. Then monocyte. Monocytes are the largest white blood cells and they are having a kidney-shaped nucleus with abundant cytoplasm. The main function is they are important in the process of phagocytosis. They differentiate into macrophages in the tissues and they also act as an antigen presenting cell. Platelets, they are also known as thrombocytes. They are small, anucleated cytoplasmic fragments that are derived from a larger cell that are known as megakaryocytes. They are primarily involved in the process of blood clotting. They are small, disc-like at the resting state, while they become irregular with pseudopodia when are activated. They are very small and their normal count ranges from 1,50,000 to 4 lakh per microliter, and the lifespan is about 7 to 2 days, and they are removed mainly by the spleen. Now coming to the plasma component, right? Up till now we have discussed the cellular component that include the red blood cells, the white blood cells, the different type of white blood cells and the platelet. Now coming to the plasma. Plasma is a clear, straw-colored fluid in which the blood cells are suspended. It's mostly composed of water, 91 to 92%, and remaining is the solid material. Solid components include inorganic constituents, mainly electrolytes that include sodium, potassium, calcium, magnesium, chloride, bicarb, phosphate, etc. While organic constituents include proteins like albumin, globulin, fibrinogen, prothrombin, etc. and nitrogenous product like urea, uric acid, creatinine, ammonia, amino acid, glucose, lipids that include neutral fats, phospholipids, cholesterol ether, etc. Other substances like enzymes, hormones, pigment like bilirubin and carotene are also present in the plasma. Now let's move on to the plasma protein. They are defined as proteins that are present in the plasma, mainly synthesized in the liver. Their main function include they are responsible for the maintenance of osmotic balance. They are responsible for the transportation of the substance like hormones, like other different type of substances. They are also important in the blood coagulation. Plasma protein constitute about 6 to 8 gram per deciliter of the plasma, and they are primarily classified into three main types. Albumin, that is the main protein responsible to maintain oncotic pressure and the transportation of other proteins. Globulins, that are important in the immunity and are also responsible for the transportation of other substances, as well as fibrinogen and other clotting factors that are involved in clotting and hemostasis. Now let's focus on clotting factors. Plasma proteins that circulate in an inactive form and then they are sequentially activated to bring about blood coagulation are known as clotting factors. They are numbered from 1 to 13 in the Roman number. Most clotting factors are plasma protein except factor 3, that is tissue factor, that is derived from the tissues, and factor 4, that is calcium, is not a protein. Okay, this makes a very important viva and MCQ, that what clotting factors are not protein. So majority of these clotting factors are again synthesized in the liver, and vitamin K dependent factor include factor number 2, 7, 9 and 10. This clotting happens by three different type of clotting pathways. They include intrinsic pathway, extrinsic pathway and the common pathway. We will discuss more about this clotting cascade in our another video. Now let's discuss the clotting factors. On the left side you will find a list of factor numbers in the Roman numbers, and here you will find the name of each clotting factor along with their function. Just take an example of factor number 1, it's fibrinogen. It's converted to fibrin to form a clot mesh that is very important for the process of clotting. Factor 2, prothrombin, it's converted to thrombin and responsible for the activation of fibrin. Third is the tissue factor, that is also known as thromboplastin. It initiates the extrinsic pathway. Number 4, calcium. It's a cofactor in multiple steps for the coagulation. Then we have 5, that is proaccelerin or labile factor. It's a cofactor for prothrombin activation. Then we have factor 7, factor 8, factor 9, factor 10, factor 11, 12 and 13. They all factors are very important and you have to remember their functions along with their Roman number. Okay, and this forms a very important bulk of the MCQs and even as the viva questions, right? Okay, for example, if I ask you that what are the factors that convert prothrombin to thrombin, then what will be your answer? Yes, they include factor number 10 mainly, right? So that's how you have to plan and prepare your viva. Have you noticed here something? Can you tell me if there is something? Yes, you are very right because there is no factor 6 mentioned here. So actually the thing is that historically factor 6 was described as accelerin. It is actually the activated form of factor 5, right? It's not a separate independent clotting factor. So this, to avoid this confusion and duplication, factor 6 was removed from the classification and now factor 5 alone is used with its activated form as factor 5. Okay, so that's why the factor 6 is not there and has been obsolete from this factor list. Now coming to the functions of the blood. This is the most important slide with regards to your viva and even your MCQs. Functions of the blood include transportation function, regulatory functions, defensive function, excretory function, and function of the blood as a buffer, right? So just remember these main headings and then you will automatically recall the main important functions. For example, in the transport function, blood is responsible for the transportation of respiratory gases with the help of red blood cells, and the gases could be any gas like carbon dioxide, oxygen, etc. Blood is responsible for the transportation of the nutrient through plasma. Hormone, vitamin and other substances, they are also transported in the blood with the help of plasma and with other plasma proteins. Metabolic waste products, they are also being carried by the blood. Regarding regulatory functions, it's responsible for the maintenance of fluid balance, acid-base balance, because it contains important buffer system like hemoglobin, carbonic anhydrase, and several type of plasma proteins. It is responsible for the maintenance of a normal body temperature. It maintains the colloid osmotic pressure, and it is mainly maintained by the plasma protein. It also responsible for the maintenance of blood pressure. Regarding defensive function, as we have just discussed, it provides protection against the infections with the help of white blood cells, prevention of blood loss with the help of platelets. Excretory function, blood is responsible for the removal of urea, creatinine, and bilirubin through the blood. And then buffer system, it includes hemoglobin as the most important buffer and the plasma protein. So this is all about the major function of the blood. Now, so today we have covered the blood as a complete functional system, not just a fluid but a highly organized tissue that is essential for our life. So let's quickly revise the composition. Plasma and formed elements, the key components that include red blood cells, white blood cells, platelets, plasma proteins, and the clotting factors. Main functions: transportation, regulation, protection, and the maintenance of homeostasis. If you understand how each component contribute, you have already built the foundation for hematology, pathology, and the clinical medicine. So before we end, a quick question for you. Which plasma protein is most important for the maintenance of oncotic pressure? I am repeating again, which plasma protein is most important for maintaining oncotic pressure? So write your answer in the comments. I will check them. And if this helped you, make sure to like, share, and subscribe for more clear exam focused physiology. Thank you so much. Allah Hafiz.